Okay, chapter 15, the chemical senses. So the chemical senses are, well, the first reason we evolve chemical senses is really to sort of be gatekeepers to our body. Our chemical senses are there to help us to identify things that we can eat safely and things that we should avoid that might make us sick or kill us. Things that should be rejected because they're harmful. And they, so since they're so related to survival, things like taste and smell can cause some very strong affective responses. So they can either cause very pleasurable responses, such as when you eat something that's really tasty and when you're really hungry, or they can cause some very nasty averse responses, such as when you smell something dying or smell a chemical that could harm you. So the taste system is the first chemical sense we're going to talk about is taste. So the taste system has evolved in humans to detect a number of different what we refer to as flavors, of course. Sweetness is usually associated with substances that have a nutritive value, that have some like high calories, for example, like sugar and carbohydrates, right, which are really yummy, right? Bitter is usually associated with substances that can be harmful, poisons, and other things that you really shouldn't that might harm your insides. Salty is there to indicate the presence of sodium, which we need to survive. We need to have a certain amount of sodium in our diet to survive, and salty gives us that flavor. But here's the thing about taste, and here's the thing you need to remember as we learn about taste and even smell, is there's not a perfect connection between the tastes and the functions of substances. In other words, just because something tastes a certain way doesn't mean that it is intended to be good for you or it is intended to be bad for you. The relationship between the taste and what substances do to our bodies is much more complicated than that. So these are typically the 5 basic taste qualities that have been identified by the research. We have salty. So you got your salty snacks like potato chips and things like that. Sour, like sour fruits and those awful sour candies that my kids like to eat. Sweet, which is awesome, right? Sweet is also related to carb as well. So that's sort of like that carb sweetie taste. Bitter, which is related to certain chemicals and things we shouldn't eat. And one that's been identified in the last 25 years or so, umami, which is the meaty, brothy taste. And it's best represented by the compound monosodium glutamate, MSG. So those are your 5 basic taste qualities. And what you can do is you can give someone a chemical and then say, okay, along the dimensions of salty, sour, sweet, and bitter, how much, what's the magnitude of each one of these dimensions for that particular chemical? So, for example, potassium chloride, which is the KCl chemical that you see here on this slide. Potassium chloride tastes salty, right? So it's like sodium chloride, which is salt, table salt, right? So potassium chloride tastes very salty. So you'll see that the salty line, which is there on the left, is much higher than the sour line, sweet line, or bitter line for potassium chloride. However, for sodium nitrate, NaNO3, you'll see that it's kind of salty, it's also kind of sour, and it's also bitter, but it's not very sweet, as you can see there by the flavor profile there. So in this way, we can start to get some basic, collect some basic data, some information on how our taste system is processing certain chemicals, how we're perceiving certain chemicals. So the way the taste system is set up, of course, it's we do a lot of our tasting, not all of our tasting, but most of our tasting is done with the tongue. Right? So the tongue contains these little bumps on it called papillae. And there are 4 different types of papillae that are listed here on this slide. The filiform, which are kind of shaped like cones. So they're kind of like that, like little mountains. And they're located all over the whole surface of the tongue. The fungiform papillae are sort of shaped like little mushrooms. That's why they're called fungiform. And they're found on the sides of the tongue and on the tip of the tongue. Foliate papillae are located in a series of folds that are on the back of the tongue and the sides of the tongue. And then the circumvallate papillae are shaped like flat little mounds. So it's like a little mound, but it's kind of flat at the top. So like if you took a little mountain and cut the top off of it, those are circumvallate papillae, and they're located in a little trench that's in the back of your tongue. Right, so filiform, fungiform, foliate, and circumvallate papillae. Inside the papillae, inside each one are these little organelles called taste buds. The little taste buds are located in all the papillae except for the filiform. If you remember, the filiform are the ones that are shaped like cones, are located over the entire surface. But all of these other papillae here, the fungiform, foliate, and circumvallate, they all have these little taste buds. Altogether, there's about roughly in the human tongue, there's about 10,000 taste buds. Each individual taste bud has about 50 to 100 taste cells in it. And the taste cells are the actual cells that do the transduction from the chemicals that go into the mouth and turn those chemical stimuli into signals that then go off into the central nervous system. So the taste buds have 50 to 100 taste cells, and the tips of those taste cells extend into the little pore, which is inside the papillae. So each papillae has a little pit in it. Each taste bud has a little pit in it. And the taste cell extends in there, and stuff gets in there and stimulates the taste cell. The transduction occurs, as I said, when the chemicals that are in whatever you're eating contact the receptor sites on those tips. So those tips contain molecules, proteins, that do different things based on the kind of flavor that they're tasting. So here's a little cartoon drawing. If you go up to the top there, in the upper left, you see a big old nasty tongue there. There's the circumvallate papillae, the foliate papillae, the filiform papillae, and the fungiform all around the tongue. In part B there of this diagram, you see a close-up of the fungiform papillae, and inside of the papillae you see those little spidery things there. Those are taste buds, right? And then in C we have a close-up of an actual taste bud, and notice that the taste bud is built up. Looks kind of like an orange, right? That's a peeled orange, right? And each one of the slices of the orange is a taste cell that is connected to nerve fibers. If we do a close-up there of the very tips of one of the taste cells, at the very top you see the very tip. It's got the little those little hairs on it, those little extensions on it, and inside those extensions are the part that feed into the taste pore and receive the chemical that it is you happen to be tasting. So some of these extensions may have receptors for bitter, as you can see there in part E of this slide. Receptors for bitter, receptors for sweet, which are activated when they come into contact with a particular chemical. Sour gets activated when hydrogen, free hydrogen that's being released from whatever you're eating, passes through a certain protein channel. And then, of course, salt happens, the experience of salt happens when sodium passes through a particular channel. All of these different chemicals come into contact with receptors or channels in the at the tips of the taste cells and activate the taste cell, which then sends its output to the central nervous system through those nerve fibers. So here, table 15.1 has a good summary of structures that are in the taste system. You have your tongue, the receptor sheet for taste, so that's the main receptor for taste. Has all the structures, including the papillae, that you need to taste. The papillae themselves are the structures that give the tongue its rough appearance. So the reason your tongue looks rough is because of the papillae. And there's 4 kinds that you have to know about. Inside the papillae are taste buds. There's about 10,000 of those. The taste buds contain the taste cells that make up a taste bud. And there's a lot of different cells for each bud, and the tip of each one sticks into the pore. The receptor sites at the tips of each of the taste cells are the sites that respond to different chemicals based on which receptor or which ion channel, like sodium or hydrogen, happens to be located on the tips of each taste cell. So here's a nasty picture here of a tongue. And those are your little papillae. And if you get a really close-up view of your tongue, you can see the ones on your papillae too. I won't show you my tongue because I have cracks in my tongue, so it looks kind of creepy. So it creeps my kids out. So it'll probably creep you out too. So the signals from the taste cells. All right. So once the taste cells are activated with whatever, right, sour, salty, sweet, whatever. Once they get activated, those signals travel from the taste cells, and they travel along a set of very well identified pathways. There's a nerve called the chorda tympani, which and this starts from the front and the sides of the tongue. The glossopharyngeal nerve, one of the cranial nerves, travels from the back of the tongue. The vagus nerve, which is a very important nerve that runs all the way from your gut all the way into your brain, but your vagus nerve catches some inputs from the mouth and the throat. And then the superficial petronasal nerve from the soft palate. The soft palate is the back upper part of your mouth, that right there in between your nose and your mouth. If you snore like I do, your soft palate, that's what vibrates when you snore, is your soft palate. But there's a nerve, the superficial petronasal nerve, that emerges from the soft palate and transmits some taste information. The pathways, all these different pathways make connections to the nucleus of the solitary tract, the NST, nucleus of the solitary tract, which is way down in your brain. The slide here says spinal cord. And it's basically the top of your spinal cord, right where your spinal cord sort of starts to become your brain stem. It's way down in your spinal cord, which tells us that from an evolutionary perspective, this is a very ancient part of our brain. This taste, the mechanisms of taste, are based upon very ancient, old connections, old structures evolutionarily. So from the NST, from the nucleus of the solitary tract, they go to the thalamus. If you remember, the thalamus is that large subcortical structure that we have in our brains. We have one on each side of our brains, and that's responsible for integrating sensations. So like all the other senses that we've studied this semester, the taste goes into your thalamus, and then from there projects out to areas in the frontal lobe. The insula, the frontal operculum cortex, and the orbital frontal cortex. All of these areas receive ultimately the input of information about the taste that you're perceiving. So here's a cartoon drawing of these connections that we just talked about. So if you look down at the very bottom, right there, right in the brain stem, where the nucleus of the solitary tract is, and we're looking at this person's brain from behind. So a chunk of their brain has been sliced off like this, and their cerebellum has been sliced off, so we can just kind of see the back a little bit. So in the nucleus of the solitary tract, that's this spot here. And then all these projections from the NST go up into the thalamus here, and then from the thalamus they project into the insula and different parts of the cortex. The insula, in particular, has been implicated in our emotional processing, our ability to process feelings and emotions. So, emotional cognition, I guess. So it makes sense that a lot of our taste information goes to an area that's related to the emotional experience of things.